So these glycoproteins, they hold themselves embedded into the membrane of the host cell, and then this capsosome, the capsid consisting of the capsosome starts to break apart, and then it releases genetic material in through the host cell's membrane. An example of an envelope virus is that of the influenza virus, which you can see here, and this too is fitted with glycoproteins, which are used for attachment purposes. And then again, you have the uncoating of the bacteria once it adheres to a cell, a virus once it adheres to a cell, and then it releases the genetic material down into the cell for infection purposes. And then we also have the bacteriophage, which is kind of a common viral shape to see. You have DNA that's situated in the head of this particular bacteriophage, and the head then is connected to something called the sheath, and the sheath is affixed to these tail fibers that are used for attachment. So very much like a syringe, but the DNA is passed through the head, passed through the sheath, and all of this makes up the capsid, and then it passes down through the cell membrane of the host cell to be infected. So essentially we have 4 classifications of viruses, and these are pretty much determinant primarily on nucleic acids, and we're still learning all we can about viruses. There's a good number of viruses out there that we're still researching to further understand the classification system. But most genomes from a virus that are going to be pretty significant are double-stranded DNA viruses, and we also have double-stranded RNA viruses, as well as single-stranded RNA viruses and single-stranded DNA viruses. So the double-stranded nature basically just means that you have 2 copies of the DNA, or the DNA is double-stranded. Same thing with the RNA. So there's viruses that will contain 2 strands of DNA, which are called double-stranded DNA viruses. There are viruses that contain 2 strands of RNA, which are considered double-stranded viruses. And then you have the 2 single strands. You can have a single-stranded RNA virus or a single-stranded DNA virus. And the genomes of these viruses are typically in circular formation, but there are many of them that are also going to be in linear formation or linear structure. So here's a cool little kind of breakdown of DNA virus and RNA virus. So again, single-stranded DNA or double-stranded DNA, and you can also have circular DNA. For RNA, you can have double-stranded DNA, and then that double-stranded DNA can be segmented or sectioned off into different segments. You can have a single-strand RNA, which is going to be fractionated as well. And then you can also have a positive sense RNA or a negative sense RNA that will also fit the scheme. Here's an image of an interesting kind of view of the herpes virus as it's essentially making envelopes that are going to appear once the virus has left a host cell through the process of budding. And so what you can see in this figure, if you look at pictures 1, 2, and 3, is that you'll actually see the virus situated inside the nucleus. So look at those. So 1, 2, and 3, the virus is situated inside of them. And then in picture 4, or at least the number 4 on the diagram, you can see the virus is starting to leave the nucleus through the process of budding. So if you look at picture 4 here, you can kind of see that process taking effect on the outside of the membrane. And then in the pictures 5 and 6, these are viruses that have already developed the envelopes surrounding them. And that's what you're able to see here in this picture of both 5 and 6. And so as this process is happening, you can end up with this particular end result where you have a pimple-like structures which are situated on the outside of certain areas of the body. So you can have this in the oral area of the body. You can also have it in the vaginal or genitalia region of the male anatomy. And just so you know a little bit about the herpes virus, this is a virus that is actually an envelope virus, and it has a polyhedral envelope to it. But the DNA type that's inside the virus is going to be double-stranded DNA. And there's a couple different types of herpes virus that are out there. There's simplex 1, and simplex 1 is what we're looking at right here, where you simply have some fever blisters or some cold sores that are going to exist in some areas of the body, primarily around the oral cavity, as I said. There's also the herpes simplex 2 virus, which is what you would have if you had genital herpes. There's herpes zoster virus, which is going to lead to the development of shingles, and then there's also the varicella virus, which is also known as chickenpox. And so mostly everybody in this course has probably had chickenpox before, so you've been infected with a herpes virus. And the scientific name is called varicella. Okay, so let's move on to some different sizes of the virions, as well as relation to nucleic acids and bacteria. So if you look at this particular diagram, you can see that these viruses are just all lots smaller than bacteria, as we have talked about in here before. But there's a good variation of the different sizes of viruses that are out there. Because remember, these are just so incredibly tiny, we have to use specialized microscopes in order to view them. So we have vaccinovirus, we have chlamydia, which is a bacteria. We have bacteriophage, which is almost looks like in this diagram the size of chlamydia. And tobacco mosaic virus, more bacteriophages, and all these teeny tiny viruses that exist. The vaccinia virus, as you can see, is a bit larger than the chlamydia bacteria, primarily just to show you the range in terms of the size and comparison of what these virions are like. So depending on the bacteria type, you may have some that are smaller than specific viruses, like the vaccinia virus. Okay, so we're going to talk about the different types of viruses that are out there. So bacteriophage is an interesting type of virus because it has a very unique structure to it. And bacteriophage is a virus that is, as the name implies, going to infect bacteria. Another kind of short term for these particular types of viruses are known as phages. And there's two types. We have virulent bacteriophages, and then we have temperate bacteriophages. Virulent bacteriophages are going to cause the initiation of something called the lytic cycle, which we have talked about before when we talked about viral replication. And with a lytic cycle, you essentially have a virus inject its DNA into a host cell, such as the bacteria, and then the bacteria will start to replicate the proteins and genetic material of the bacteriophage that's injected itself in its genetic material into the host cell. And when that takes place, then you essentially have the destruction of the bacterial cell once that bacteriophage tries to leave the host cell. So you have complete destruction of that. And there's 5 steps that are used by viruses to allow this process of replication to take place. And those steps are called attachment, penetration, biosynthesis, assembly, and release. With the process of attachment, you essentially have this feature here. You have here a cell that's known as Vibrio cholerae, which is an organism that's going to cause cholera, or responsible for the cholera outbreak. And this organism is being attacked by a virion known as CPT1. The CPT1 virus is going to start attaching itself to the Vibrio cholera cell, and once the attachment has happened, then you have the process of penetration, where the virus will then act like a syringe, and it's going to pass this genetic material through the sheath of the CPT1 virus, and once it's passed through, then you have the process of penetration, and then that genetic material will integrate into the cholera cell, and it will undergo the process of biosynthesis, where you have replication, transcription and translation of the genetic material of the virus, as well as proteins in the virus. So those things will start to be biosynthesized. And then after that process takes place and you have the synthesis of the viral components, then you have to have assembly of them. And assembly means that inside of the cell you're going to develop new bacteriophages. So the head of the bacteriophage is going to develop, the tail fibers of the bacteriophage, and the sheath will develop. And they'll all come together to formulate new viruses. So we call that assembly, where we have the assembly of viruses. And then after assembly takes place, you have the process of release, where the cell here in this picture will become overwhelmed with the components created inside of it, of the virus. And once creation takes place, all of those virus particles will start to bud through the cell, weakening the cell membrane and causing lysis of the cell. So this whole process is known as the lytic cycle because it's basically lethal to the cell, and it will cause destruction of the cell. Okay, so that's kind of how the lytic cycle works. And here's kind of an outline of what the bacteriophage looks like, a little bit more up close and a little bit clearly defined in terms of the structures. You have this head portion, which we talked about before, which typically has 20 facets on it. And each of those facets are filled with genetic material. So the head is comprised of the capsid that will surround the genetic material. And then you have this sheath that the genetic material is going to pass through. So that one sheath end is known as the collar, the other end is called the end plate. The genetic material is going to pass straight through the sheath, and then it will be injected down into the host cell that's going to be impacted. And so the bacteriophage is able to undergo this process because it uses these tail fibers to allow it to dock or adhere to the cell membrane of the host cell being infected. And since this is a bacteriophage, it's anchoring itself to the cell wall of the bacteria. And in this image, like I said, this is a cholera cell in which this this event is taking place. Okay, so that's a little bit about bacteriophages and their role in with the lytic cycle. Again, with viruses, you have attachment and penetration, which are kind of the key things that take place when you have a virus infecting a cell. With animal viruses, we have some primary steps that take place, and for the most part, these are all exactly the same as what we saw with the lytic cycle: attachment, penetrate, biosynthesize, assembly, and release. Pretty much the same steps here for the animal virus. If viruses need to replicate themselves, they're going to attach to the cell. They'll penetrate down in the cell. Then what they will do is do a process called uncoating, and this is where the capsid is going to start